Hello, my name is Maninder Ahluwalia. I am from Cardiff Metropolitan University, and in this lecture today, I'll be talking about gene therapy. But before I talk about the gene therapy, let's have a look at what are the genes and why do we need to do gene therapy if we have to. Looking at the adult human body, we have about 75 to 100 trillion cells in our body. And in each of these cells, except the sperms and eggs, we have 46 chromosomes. Within these chromosomes, we have about 1.8 meter long DNA in each cell. So the purpose of this DNA is that we have about 20 to 21,000 genes. And genes are segment of DNA that has information to make protein. Before this gene is made into proteins, there is an intermediary step, and the molecule there that is made is called a messenger RNA. And this messenger RNA then gets out of the nucleus, gets converted into protein. Proteins are synthesized, and proteins are responsible for all the functions which are performed in a cell or a body. So if you think of any function a cell is performing, there will be a protein responsible for that. So I am going to talk about this part of this diagram, the genes, and what is the purpose of it and how do we maintain it. And if you look at the structural level of the DNA or the structure of the gene, we are 99.9% identical with each other, but we are still unique in certain ways. And these are the certain polymorphisms that we call, or you can think of them as spelling errors within our genome that make us unique. So these spelling errors that exist are responsible for the 0.1% difference between individuals, could mean that the cell may or may not be able to perform the function it is required to do. So if we have a normal gene here, that makes a functional normal protein, that is fine. But sometimes what can happen is there could be a mutation. There could be a spelling error. That could mean that either there will be no protein coming off that gene, or the protein that is being made is abnormal and it can't perform the function that it was supposed to perform. And if you think of in a cell, so I have a gene here, which is a normal gene. Through the steps of messenger RNA and making into protein, we have a functional protein here. If you think of this protein as being an enzyme, which is required to convert this substrate into the product, and the gene is normal, the functional enzyme is being synthesized, the substrate will be converted into product, and the metabolism or this reaction can happen normally in the cell. But what if there is a mutation in that gene? What if there is a spelling error? And that could mean either there is no protein that is being synthesized, or a different type of protein is being synthesized, which is not capable of performing the function. So the substrate here will not be converted into product. And if you think of the functioning of the cell, and in this case, it is possible that the cell will accumulate this substrate, and that could be toxic to the cell. Or there may be no product being formed, and that could mean that the cell is depleted of this product, which is required for the functioning of the cell. So if there is slight variation in the gene, that could mean that this metabolic process may not happen as we want it to happen. So that is about the structure of the DNA. And I also mentioned that this DNA makes protein. So the process of making DNA into protein, we also call it gene expression. So if we say gene is switched on, gene is making protein, and we say gene is expressed. And not all the genes are expressed at the same level. So here I have an example of protein A and protein B. And protein A is highly expressed, whereas protein B level is at a low level of expression. And this expression is also responsible for the normal functioning of the cell. So if the cell needs to metabolize something, and that metabolizing enzyme is this protein A, we need to make multiple copies of that. So this expression is also important for the normal functioning of the cell. And for this gene to be expressed to make protein or being switched on, we need certain other proteins to function here. So within the nucleus of the cell, and here I have the gene, that is the segment of DNA that is going to give me protein. But there is also another region within our genome, which we call promoter, which is generally quite close to the gene. And certain transcription factors, certain proteins have to come and bind here. And if these proteins come and bind here, then only that gene will be switched on. So it is the availability of these transcription factors. It is the structure of this promoter that is also responsible whether the gene will be switched on or not, whether the protein will be made from that gene or not. Let's look at an example. So when we eat, the excess glucose can be converted into glucose 6-phosphate, into glucose 1-phosphate, and then into glycogen. Glycogen can be stored in the liver. And at the time of, during the times of fasting, this glycogen can be converted back into glucose, and glucose is used to make energy for survival. So that is normally what happens. We can store glucose in the form of glycogen, and at the time of need, we can break down glycogen to form glucose. And there's an enzyme called glucose 6-phosphatase that converts glucose 6-phosphate into glucose. This enzyme is a protein, and the information to make this protein is in this gene, glucose 6-phosphatase, which is present on chromosome number 17 in our cells. And we have 2 copies of chromosome 17, one maternal and one paternal. They are identical, and they both have this gene. We need 2 functional copies of this gene for this metabolism to carry on happening. What happens in case of this disease called glycogen storage disease 1A, that there is a mutation in glucose 6-phosphatase. That means if both the chromosomes have got this spelling error, there is not going to be enough glucose 6-phosphatase present. So the protein that is coming off is non-functional. That means glycogen cannot be broken down into glucose. And that's what happened in this disease. That means there are severe and life-threatening episodes of hypoglycemia, low glucose level, which can lead to long-term diseases like liver and kidney diseases. With other multiple factors, it leads to liver adenomas and carcinomas. And at the moment, it is being managed by dietary measures, repeated consumption of cornstarch, and the ultimate, what we need is liver transplant. So this mutation, this spelling error, that means that we have this disease. Glycogen is being stored in the liver and it's not being broken down into glucose. So that is genetic level error. Can we repair it? And it's been found out that we only need little bit of activity of this gene to make enough protein and to avoid these liver cancers stage progression. So we have 2 chromosomes, maternal and paternal, with mutations on glucose 6-phosphatase gene. Can we just change that gene? Can we just put a new version of the correct version of the gene, even a little bit, to revert the cell for making this glucose 6-phosphatase to enough level that glycogen can be broken down? So that is an example of a disease. And now that takes me to the topic of gene therapy. So if we look at the definition of gene therapy, the gene therapy is being defined as introduction, removal, or changing the content of the genetic component of the individual in the cell with the aim to treat or cure a disease. So we want to put a new gene into the cell. And I'll talk about which type of genes in a minute and how we put them in. But the ultimate aim is we want to have durable expression of the gene that we want the cell to have at a sufficient level. So gene therapy is introduction, expression of a therapeutic gene which will be expressed in the target cell that we want it for. We sometimes want to express the gene, so we want to add the gene which will make the protein, and we call it as gene augmentation therapy. So here we have a cell with a non-functioning gene where we can put a functioning copy of the gene and the cell will start to make normal protein and carry on functioning normally. And that you can think of this glycogen storage disease that I mentioned, where we don't have that enzyme being formed. Can we put a copy of the glucose 6-phosphatase gene there so that this breaking down of glycogen can happen to glucose? Other thing is sometimes we have certain proteins which are toxic to the cells, and we don't want those proteins to be made in the cell. And in that case, we can put some gene that will block the synthesis of that toxic protein. So this red gene here is toxic to the cell. It's faulty. We don't want it to make protein. We can put this blocking gene into the cell, which will block the synthesis of this toxic protein, and cell can carry on functioning normally. So we call that therapy or gene therapy as gene silencing therapy. And sometimes we may want to kill the cell. I know killing the cell is not one of the things that you want to do, but sometimes killing the cell is important. And I want you to think about a cancer cell. And in that case, we may want the cell to die. In that case, we can put a suicide gene, which will make a protein that is toxic and it will cause the cell death. Sometimes we want to train our cells to kill one particular type of cell. So we may want one particular type of cell not to exist. But how do we get rid of that? We can put a gene there so that it will start to make that protein that the cell can then, our immune cells can recognize and say, okay, we want to get rid of this cell, and those cells can be removed. So this marker gene is being introduced to the cell. Cell starts to express that marker on the cell surface. Immune cells recognize that marker and they come and attack this cell and the cell dies. So we can use gene therapy to kill specific type of cells. So that is what we want from gene therapy. We want to cure a disease by altering the genetic structure of the cell. How do we do that? There are two types of approaches that have been used. One is in vivo, where we put the gene that we want the body to have directly into the body. And that is called in vivo gene therapy. So we use different measures, and I'll talk about that in a minute. We put them directly into the body, and that is called in vivo gene therapy. Other approach that has been used is that we can take patient cells out of patient. We can repair those cells with gene therapy and then reinfuse those cells into the body. That approach is called ex vivo. So you take cells out and then manipulate them and then put the corrected version of cells into the body. So there are two approaches at the organismal level, but how do we put this DNA into the cell? Remember that DNA is a big molecule. It is charged molecule, and cell is not just going to pick up the naked DNA into the cell. So we have to trick the cell, and we need to package this cell, hide it in a vector. So we need to put this DNA or the gene into a vector so that the vector can go into the cell. Two types of vectors have been used, non-viral vectors and viral vectors. And as the name suggests, non-viral vectors don't use any viruses. Here we are hiding this gene in some particles, which can be small nanoparticles or polymers or lipid kind of bilayers. Basically, we want to trick the cells to let them into the cell. Once they go into the cell, they are carrying our cargo, the gene we want to go into the cell. So that is actually hidden there. We trick the cells to have them, and this DNA from these cells go into the nucleus, and that's what we want. That is our aim. They don't cause any immune reaction. They are safe to produce. You can produce them in bulk, but cell doesn't take them up as much as we want them to. So in the last decade, about 15 to 20% of the gene therapies that have been developed have used non-viral vectors. Most of these therapies that have been trialed use the viral vectors. And as the name suggests, we use viruses in this case. Viruses have kind of inherent capacity that they can get into our cells, and they are very good at taking their genome and putting their genome into our cell. There are different types of viruses. So when they go into our cell, the DNA or the RNA that they are carrying, so they have genetic material. Ultimately, that genetic material gets into the nucleus and start to make proteins using the machinery that is already present in the cell. So those are called viral vectors, and the technology is much more advanced in case of viral vectors as compared to non-viral vectors. And in the last decade, about 70 to 80% of the therapies which have been trialed use the viral vectors. So I'm going to talk about these viral vectors because these are being developed and have been used for clinical purposes. Looking at the virus has got an outer protein coating. It's a capsid with the proteins. And within that, there is the viral genome. This viral genome gets into the nucleus. And if we take a virus, put it on a cell, that cell will be infected with this genome. This genome will make more of its own copies, make viral proteins, and that cell will shed that virus particles again. So you infect one type of cells with virus, you make more viruses. Or you don't even have to put the virus, you can take the viral genome, hide it in some sort of circular DNA, which we call plasmid, put that into the cell, and still the cell will make virus particles from there. Now we want to use this ability of the virus of infecting a cell and making more viruses. We don't want to make actually virus. We want to have virus with the gene we want cell to express. So what we can do is we can split the genome of this virus, also put the genome of the gene that we want to express, use the same cell, which will make viruses, but this virus now will have the protein that we want the cell to be infected with. So these viruses which are used are of different types. The most commonly used are the adeno-associated viruses, adenoviruses, and these lentiviruses. I don't want you to pay too much attention into the types of viruses, just that they have different type of the DNA or RNA, so the genome could be different. The size of genome is different. The type of immune reaction they produce in the host can be different. And the size of the DNA that they can carry is different. But I'm not going to talk that much about the nitty-gritty of the different types of viruses, but I'm going to talk about how do we trick this virus into having the protein that we want the cell to have. So here is my viral genome. It's got different components. It has got components which is required for virus replication and virus structure. And it has got this purple rectangle here. These are the genes which are responsible for the pathogenicity or the disease causing ability from that virus. So that is our natural virus genome. What we do now is we split the virus genome into two parts. We take the replication and viral structural genes and have them separate. And then we remove this disease causing gene from the viral genome. This purple is removed, and instead of that, we put the gene that we want the cell to have. So this is the transgene or the therapeutic gene that we want to send to the cell. So we have two constructs, packaging construct, which will make the virus, the empty virus. And then we have this viral vector construct where we have the gene that we want to send into the cell. We put these two things into a packaging cell. So these are human cells which we can use as kind of factories to produce more viruses. We put these two constructs into the packaging cell. The packaging cell makes the viral proteins and the genes that we want to insert and sheds virus, which is now recombinant. So now we have virus here, which is got the gene of interest that I wanted to send to the cell. So what I've done is I have split the genome of the virus, removed the disease causing gene, put the therapeutic gene, and now I have produced virus particles with the gene I want. We collect those recombinant virus particles and we put them on the target cell. So this is the target cell now. I want to send this particular gene into the cell. So these are the viruses which I produced. I put them on my target cell. Target cells take these viruses thinking it's a virus, and virus will get into the cell. And then this genome gets into the nucleus. And here we have our chromosomal DNA. Depending on the type of virus, remember I was talking about different types. Some of the viral genome can get integrated into the chromosome, or some of the different types of viruses stay on their own. We call them episomal DNA. In either case, they both will be made into proteins, and this protein is what we wanted the cell to make, because cell was not making this protein. And depending on the type of cell, we may want to choose the different types of viruses. And the ultimate aim is that the target cell starts to express this therapeutic gene that we want. So we can do that. We can manipulate and we can use viruses, and we can put the gene that we want via viruses into the cell. Most commonly used virus in clinical trials right now is this AAV, Adeno-associated virus. It has got very low immunogenicity, so that is not causing our immune cells, it's not activating it as much, and it's being used for various clinical trials. So in any case, so we have, again, we split the genome, we put it into the packaging cell, and we have the virus particle. Virus particle is taken up by the target cell, which enters the nucleus, which introduces its DNA or RNA into the cell. Either can get integrated into the chromosome. It doesn't happen as much with AAV virus, or stays as episome. which gets converted into the messenger RNA. Remember that messenger RNA is the intermediary step before the proteins are made, and the proteins are going to perform the function or go out of the cell and perform the function. So in theory, those are the basic steps of making a cell to start making protein that it was deficient of in the first stage. But there are various challenges. First is, how do we deliver these things? How do we deliver efficiently? And how do we deliver to the right type of cell? So there are various strategies which are being developed to send the gene to the right type of cell. Then we also want to have expression. So there is no point putting the gene if it is not going to make the protein. The other concept is that how do we maintain? We give the gene therapy once, and that has to be good enough to maintain the expression level long term. One of the main concerns is we don't want to disrupt any other gene. We don't want to cause problems with rest of the genome. So we don't want to disrupt any other genes, neighboring or otherwise. We also want to fool our immune system. We don't want the immune system to start to attack those cells and get rid of them before they can make the functional protein. So those are the things. And there is another concept that we also need to think, how much is it going to cost? It is taking quite a lot of effort and money into producing one gene therapy, but there are certain diseases where it is showing that long-term annual cost of maintaining somebody on a disease and one-time cost of gene therapy could cancel out, but we are not there yet. So now I've got a couple of examples of gene therapy where it has been successful. And the first one is this severe combined immunodeficiency. So when you think of our immune function, we have cells, these T cells and B cells and natural killer cells in our blood. When we are exposed to any viruses or bacteria, these cells attack them and get rid of them. And we live, our body is not infected with these pathogens. And in this case of severe combined immunodeficiency, these people with the SCID, the short of this disease is called SCID, they lack kind of immune system. So they lack all type of immune protection. And there is a type of SCID called ADA-SCID, in which there is this enzyme which is called Adenosine Deaminase, which is mutated. So looking at the normal cell now, so there is a gene coding Adenosine Deaminase, which makes protein, this weird shaped protein called Adenosine Deaminase. And within our cells, there are these adenosines and deoxyadenosines, which are broken down by this enzyme called adenosine deaminase into these inosines and deoxyadenosines, which are used for purine degradation pathway, and the cell can live happily with that. So we need this enzyme to be present for the functioning of our immune cells. What happens in SCID is, or what happens in ADA-SCID is that there is a mutation here. So that means virtually no adenosine deaminase is being synthesized. Remember, this protein was required to convert adenosine into inosine. Now that is not happening. And adenosine and deoxyadenosine is toxic to the cells. So that accumulates because we can't break it down to inosine and deoxyadenosine. The cell accumulates this. And these cells, these fighter cells that our body has, they die because they go through cell death, because this is toxic when this builds up in the cells. So in this ADA-SCIDs, our T lymphocytes, B lymphocytes, and natural killing cells within blood are dead. So when we are infected, when the patient is infected with any type of pathogen, there are no fighter cells available. And these pathogens, they cause problems. So these patients, they have no immune system, and they cannot fight any type of infection. So that is ADA-SCID. Luckily, there has been one gene therapy. So basically, that gene, as I mentioned before, can we replace that gene? And this is an example of ex vivo gene therapy in case of Strimvelis. So you can take patient cells from the bone marrow or from peripheral blood. You isolate a specific type of cells expressing certain markers to indicate that this cell can make different type of cells. So these are the stem cells. So you isolate, you take my blood, you isolate CD34 cells. You grow them and you do gene therapy to them. You manipulate their genetic material. You add the gene for adenosine deaminase that was missing in ADA-SCID patients. You put that gene, now we have these cells which can make the right type of cells with expressing adenosine deaminase, and we can reinfuse those cells now into the patient. And that is the success story of Strimvelis, which is an ex vivo manipulation of cells to put the adenosine deaminase back into the patient so that they can convert adenosine to inosine and these fighter cells can survive. Another success story is for this retinal degeneration. There is a protein called RPE65. It is required for the visual processing, and it is required for survival of these photoreceptors in the retina. So here's the eye, this is the retina. Within the retina, we have these photoreceptor cells, and these cells express this protein called RPE65. If there is a mutation in RPE65, it leads to various diseases, all of them having retinal degeneration. And here, in this case, a gene therapy called Luxturna has been developed, where adeno-associated viruses have been used with the gene expressing RPE65, which can directly be injected into the retina, and these cells start to express RPE65, and the retinal degeneration can be halted or cured to some extent. I started talking about glycogen storage disease 1A. And gene therapies have been tried for this disease because remember I mentioned that you only need little bit of expression of glucose 6-phosphatase gene. Again, AAV-based gene therapy has been developed. This was the person who was given for the first time in 2018. Clinical trials have been going on. So phase 1 and phase 2 trials are showing quite positive outcomes, where 9 patients were treated and all of them are showing improved glycemic control. Some of them haven't taken any cornstarch supplementation, or some of them have reduced it by about 70%. And they are showing good levels of metabolic control and without any side effects at the moment. And currently phase 3 trials are recruiting, and we are hoping to have some outcomes, positive outcomes from this disease, gene therapy for this disease, so which is looking positive. So far I've talked about adding a gene into the cell, and I've also talked about certain challenges without going into much detail. some new approaches are coming where we are trying to figure out, rather than adding the whole gene into the cell, can we get away with by not adding the whole gene? So one of the approach currently being used is gene editing. Rather than adding the whole gene, can we just repair it? What we can do is we can put some scissors into the cell. Obviously, these are not the scissors that we would normally use. We call them molecular scissors, which will go knowing where they want to repair the defect. They go in, they make a cut, and then in that cut, we can put the right version of those few bases, and we can repair the gene. They are not being used clinically at the moment, but they are being developed as we speak. Another approach that is coming out is the messenger RNA therapy. Remember, I was talking about the gene, the DNA making protein with an intermediary molecule called messenger RNA. So here I have the AAV gene therapy that I have been talking about. We use the virus with the gene. Virus gets into the nucleus, gives the genetic material, makes the messenger RNA. Messenger RNA comes out and makes the protein. Now this therapy, as the name suggests, we don't add the whole gene. We just use the messenger RNA. Because then we don't need the messenger RNA to go into the nucleus. It goes into the cytoplasm and starts to make protein. It has multiple advantages which are being considered, but the aim is that we hide the messenger RNA in some lipid nanoparticle, let it enter the cell, and it is ready to make protein there. So it is also being developed, and one of the thing for glycogen storage disease type 1, they have trialed this messenger RNA therapy in mouse models, and they are showing some positive effects in terms of glycemic controls, even 11 days after the therapy. So it is looking promising with messenger RNA therapies. Again, they're not being currently used for clinical purposes. So if you look at the new drug developments, and this pink area is the ongoing trials, it is the number of trials using gene therapy have increased exponentially in the last decade. We still have to address certain aspects of safety, certain ethical aspects, but I think the future is looking promising. I hope you have enjoyed this talk, so thank you very much for listening.